Hello, hello. Okay, we are now recording. Can everybody hear me okay? Awesome. Okay. We'll see if more people jump in. We're just gonna jump right into it. First and foremost, hello. Sorry, it's been a little bit. We had some issues where our nanny quit and then Kate was working and all sorts of fun stuff trying to figure out school with the kid. So, but hopefully we're back and it should be consistent again at this time from here on out. So how's class going? How is biochem? Anything in particular that you'd like to talk about or to cover before we get going? Any particular questions? Certain material that maybe you've had a hard time with up to this point? Anything like that? Okay. I'll take the silence as a no. If at any point you guys do have, but I see more people are jumping in now. If there are topics that you guys have covered that are either unclear or a little bit confusing, please just either in the chat or just unmute yourself and go ahead and ask questions about that specific material, and we can go ahead and take a look at it. But I mean, really, the past couple of chapters I feel like have been relatively straightforward. Hopefully you guys feel similarly. But if not, just stop me and let me know kind of where the confusion or what your thoughts are with that. Okay. So today, kind of what I had prepared or have prepared is mostly focusing on glycolysis. So glycolysis should be your material, I believe, for this week. And it is definitely a large portion of questions from this exam and your next exam are going to be related to glycolysis and the whole process, and because there's kind of a lot to unpack with glycolysis. I'm not sure, has anybody had a chance actually to watch my recorded video that I already made covering this chapter?
I watched yours on, I think it was Tuesday. It was either Tuesday or, yeah, Tuesday.
Okay, sweet. So I'm going to want to add a couple of things into there. So I was doing a little bit of prep before before this class today, and I put together a little Word document just to kind of start going over some extra stuff and hopefully give you guys a couple of extra ways to remember a lot of this, because like I said, there's a lot of memorization for this specific unit, as well as the next unit, or the next chapter, which is the Krebs cycle. So start early with this stuff, because the questions that you're going to get, I wrote up a handful of questions that will mimic what you'll see on the exam relating to this topic. So we'll want to kind of look at that. But first things first, first thing, before anything, who can tell me what glycolysis is?
I know it's an oxidative process.
It is an oxidative process. What's the whole purpose behind glycolysis? What's it for? Why do we need glycolysis to happen? What's its goal?
Create 2-pyruvate.
Create 2-pyruvate with the very end goal of being what? Why do we go through glycolysis, why do we create the pyruvate?
For the beginning process of the Krebs cycle?
Like I said, what's the entire, so the glycolysis, the Krebs cycle, electron transport chain are all metabolic pathways, right? What is the purpose of all of the metabolic pathways? Why are they doing all of this?
To create energy.
There is to create energy. And in the human body, what is our form of energy?
ATP.
ATP. So Rebecca's on fire. Give me half a second. I need to real quick grab my baby because he's crying. Hi, bud. Come along. You. Okay, I'm back. Yeah, so the entire purpose of all of these pathways, and that's what we want to remember, the purpose is to create energy in the body. All of these pathways are necessary for us to survive and for our entire body to function and to work at all. So with glycolysis, each of these processes are going to be happening in different parts of the body. So for example, within the cell, where does glycolysis take place?
In the cytosol.
Good. In the cytosol. So glycolysis is taking place within the cytosol, which is kind of, you can think of it like the kind of the jelly that's in between everything else, is where everything is floating around inside of the cell. All of the organelles and stuff are floating inside of the cytosol. We also talk about glycolysis. Is actually it can be referred to either as an aerobic or an anaerobic metabolic process. These two words here, anaerobic and aerobic, these are the big ones that I want you to remember, because these are test questions that this word will get thrown around a lot in your exams. And if you don't know the definition of each one, and make sure that you read it carefully, it's very easy to get questions wrong because of that. So anaerobic means no oxygen present, and aerobic means oxygen is present. Please, please, please get those straight and remember those, as that will be really, really, really important, especially continuing on the end of this process, as well as the next couple of processes that we're going to look at. Okay, so those are sort of the big ideas, that glycolysis is an oxidative process, that the end goal, we're going to create 2-pyruvate, as well as some NADH and ATP. The overall purpose is to create energy. It happens in the cytosol, and it can either be an aerobic or an anaerobic metabolic process. Most of the time, we refer to glycolysis as an anaerobic process because it can still happen even without oxygen. It is the only metabolic process that can occur with no oxygen present. So looking ahead just a little bit, this right here is our glycolysis process. Starting with glucose, going all the way down, converting it into pyruvate. So what makes these next couple of units challenging is literally that it is just straight memorization. You need to know every single step, what gets converted to what the enzyme is, what is produced at every single step, as well as the order and the numbering of every single one of these steps. So it can look a little bit intimidating, but when in the YouTube video that I did, I'd take some time and I really break down kind of what each of the things are, and you can sort of follow a flow of the entire process all the way through by looking at the enzymes and understanding what the enzymes do. So we're going to start off, just going to run over these really quick with you guys. These are a couple of mnemonics that I either found or I put together hopefully to help you guys remember this. And at the end, I will go ahead and I'll save this document and I will upload it into the Teams page. So that way you guys can have access to this and pull it up and go through it yourselves, okay? So for the intermediates of glycolysis, there are technically 10 steps for the intermediates. The mnemonic that I actually found, this is an MCAT mnemonic. It's girls get fine food, gentlemen dine girls, boys prefer to pick pepperoni pizza. A lot of these mnemonics are kind of silly, but these are really good way to help you remember these sort of things. So this represents the order of everything that it goes in. It's from glucose, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, glyceraldehyde 3-phosphate, dihydroxyacetone phosphate, 1,3-biphosphoglycerate, phosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and then into our 2 pyruvates. So first, before we continue going through there, are there any general questions that people have about glycolysis or kind of how Dr. Sarkar has gone over glycolysis or any of the other information about glycolysis in general? Okay. Keep going. So that first mnemonic, it'll help a lot to just understand the intermediates. You will get asked questions about what are the like what does glucose 6-phosphate go to? What is the next step of glucose 6-phosphate? What enzyme converts glucose 6-phosphate to fructose 6-phosphate? Those are sort of the questions that you'll start to see within this material. So now looking at the enzymes, I try to combine the enzymes along with what is used up or produced during these processes. The one that I left out is actually this one, the triose phosphate isomerase, which converts DHAP, the what's it called, the dihydroxyacetone phosphate, into glyceraldehyde 3-phosphate or phosphate. So this isn't included in this mnemonic, but just remember that this is one of the steps in glycolysis. So we have Kevin Adams is crazy awesome, and Hank Nelson can absolutely make everyone Kool-Aid. Again, these are really kind of random and silly, but it's a lot easier to remember something like this than it is to remember the full name of every single one of these enzymes. So first of all, so each of these is going to stand for the letter of the type of the enzyme that it is. So all of their Ks are kinases. Who can tell me what kinase enzymes do? What's their function? What's their purpose?
Add a phosphate.
Exactly. All of our kinases add phosphate. So you can literally see glucose hexose kinase adds a phosphate. Phosphofructokinase adds a phosphate. Phosphoglycerate kinase, it just adds another phosphate. The pyruvate kinase, again, adds a phosphate to get us to pyruvate. So literally the kinases, all they do are add phosphates to what we're working with. What about isomerase? What's the function of an isomerase? Somebody besides Rebecca.
Gonna restructure the like glucose, aldehyde, fructose is ketone.
Okay, so the isomerase its function is to reconfigure a molecule. It does not change its entire structure, or it doesn't change its chemical makeup. So if you started with 5 carbons, you're still ending with 5 carbons. If you had 10 hydrogens, you're still ending with 10 hydrogens. So the chemical formula or the chemical makeup of that molecule is not changing. It's just restructuring it. So that's how we get from glucose to fructose. So when you see an isomerase, the keys are, so when you see a kinase, first of all, you know that you're going to be adding a phosphate. So then in the name, a phosphate is going to be changing. So if there's already a phosphate, it's going to be bisphosphate because now there's 2. If there wasn't a phosphate before, it's going to add 1. And the number is just going to be on which carbon that phosphate is being transplanted. And those are going to be constant. So the hexose kinase is always going to put a phosphate on the number 6 carbon of glucose. And then the phosphofructokinase is always going to put a phosphate onto the first carbon of fructose 6-phosphate. So rather than, even though you do have to really memorize and know this whole process like the back of your hand, it's still not completely memorizing so much as it is understanding each of the enzymes and their processes and kind of what they're doing. Because the enzyme itself is going to tell you what it's doing and what it's actually changing. Just like if we look at hexose kinase, the glucose, the shape of glucose is a hexagon. So hexose kinase, ose, that suffix ose is actually going to mean sugar. So glucose is a hexagonal sugar. Kinase means it gets phosphorylated. Glucose 6-phosphate, phosphoglucose isomerase, or phosphoglucose isomerase. It's a glucose that has been phosphorylated, and it's an isomerase, so it changes from glucose to fructose. So hopefully you can start to see it's not so much just memorize these random words in the same order. You can walk yourself through this entire process and even guess in naming each of these different enzymes and what their function are. Okay. Any questions about this material up to now?
None so far.
Okay. Anybody feel intimidated by glycolysis, or does it seem pretty straightforward?
Seems absolutely awful. It does, but the mnemonics are going to be helpful.
It is. So these next couple of units, the key is going to be repetition and then something called active recall. What I would actually really recommend, let me actually pull up, let me see if I can pin my, so let me grab a piece of paper real quick. The way that I studied and learned all of these processes was literally taking a piece of paper and writing out the whole process. So you'd start with glucose and literally talking yourself through the entire process. So when you start with glucose and being able to understand step by step what is actually happening. Actually, I can do this on the Word document so that way everybody can see it and I can share it with everybody later. So for example, starting with glucose and talking our way through this entire process. First off, what do we know about glucose? Where, let's say we're currently looking at this glycolysis happening inside of a muscle cell. So within a muscle cell, where is the muscle cell getting its glucose from originally? What brings it the glucose?
Blood.
Okay, so the glucose travels through the blood to get to the muscle tissue. Once it gets to the muscle tissue from the blood, how does it actually go from the blood to inside of the muscle tissue? he doesn't cover this a ton in his actual lecture, but it is in the book and in the readings, and he does mention it briefly in the lecture material as well, though, or in the vodcast and stuff. There's a special receptor that actually helps bring the glucose from outside of the cell to inside of the cell. Does anybody recall what the name of those receptors are, or the abbreviation of those receptors?
It's glut.
The glut or the GLUT receptors. Specifically, we'll mention he talks about GLUT2 in the muscle tissue. There are 4 different types of GLUT receptors, but you'll get more into that in physiology 2 in 2 trimesters when you dive a little bit deeper into insulin and glycogen and that sort of stuff. So, or glucagon and insulin, sorry. So right now we'll just talk about GLUT or GLUT2 receptors. So these receptors will help bring glucose from outside of the cell to the inside of the cell. And actually, in the process of bringing the glucose from the outside of the cell into the cell, something happens to the glucose. Who remembers what happens to the glucose?
Gains of phosphate, right? Or
Yep, it
loses
gets
one.
phosphorylated. It gains one. So as it enters the cell, glucose gets phosphorylated. So in glycolysis, in the process of glycolysis, we already talked about that a little bit. So when glucose gets phosphorylated, what's the enzyme that phosphorylates our original basic glucose?
Hexokinase.
Our hexokinase is what does it. One little note here, hexokinase is present in all of them. There is a second enzyme that is only found in the liver. I believe it is called glucokinase or something like that. I know it starts with a G.
Yeah,
I believe it's it is
power.
glucokinase. So hexokinase is in all the tissues. And then we also have glucokinase, which is in the liver. The liver, I've mentioned this before in some of my videos, and I know it's in the book a couple of times. What is different about glucose and glucose being in the liver versus it being in any other tissue? What's different about the liver? Okay, this is really good. I'm glad that we're going to cover this. So the liver, glucose can enter and leave.
I'm sorry, Drake, where are we? I'm kind of lost.
What do you mean? Like what are we talking? Like what are we talking about?
Yeah, like we're in glycosis, but in what area? Because I'm trying to follow along with the PowerPoint. Is there a
So
diagram?
I'm not necessarily going off just the PowerPoint, so because this what I'm essentially doing is going and taking all of the bits and pieces from the past couple of lessons that you have learned and merging them and combining them into one cohesive idea. Does that make sense?
Yeah, okay, I got it. I was just confused on what I was supposed to be following or something, but all right, I got it.
Yeah. So if you pull up the teams, I'm sharing my. Can everybody share the screen or see the screen that I'm sharing right now?
Yeah,
Yes.
you're doing.
Okay. Yeah. So because all of this has been covered up to this point already, or it had up to this week. It's been covered, and I'm just kind of pulling up the important and key pieces that you're going to be seeing tested on with each step of glycolysis. The very first step of the glucose entering the cells, it will be heavily tested on, which is why I'm kind of taking a moment to go over some of these details. So jumping back in, so talking about the liver is the only tissue that glucose can actually enter and leave. Any other tissue, mostly we talk about muscle, the glucose is going to be stuck. So once it enters muscle, I'm going to put a little side here. Technically, adipose tissue as well, technically, but glucose gets converted into some other stuff, which we haven't talked about quite yet. That's a couple chapters down the road. But the questions that he'll ask will include liver or mostly muscle tissue. So I'm saying that from the liver, glucose can enter and leave, and any other tissue like the muscle, especially, the glucose is going to be stuck. Once it's been phosphorylated, it is stuck inside of that tissue and cannot leave. And all of this is important. The reason why the liver, it can enter and it can leave is because this is super important when maintaining blood glucose levels. The liver is one of our key organs in maintaining our blood glucose levels to make sure that we have enough glucose in the blood at all times and that we don't get too low or we don't get too high. Okay. So this is that whole first step. So as it enters the cell, this right here is actually our first reaction. As it enters the cell, the glucose is phosphorylated by hexokinase. So glucose gets acted on by hexokinase and becomes what? What is our second intermediate in glycolysis?
Glucose 6-phosphate.
Glucose 6-phosphate. And now glucose 6-phosphate. Real quick, hexokinase. There was going to be 3 that we talk about that we have rate limiting steps or rate limiting enzymes. We talked about this way back in the enzyme chapter. I think it was chapter 2 or 3. Does anybody remember or recall what a rate limiting step or a rate limiting enzyme is or what that means? Anybody remember? Okay, so a rate limiting step or a rate limiting enzyme is exactly what it is saying in the name. It is the step or the enzyme that controls the speed or the rate at which the reaction is going to happen. Or either the reaction or the whole process is going to happen. So if we think about it, if we don't have glucose available in the blood next to that muscle tissue, and we don't have like if there's no glucose, there's none of these reactants. Hexokinase is not going to be activated, or if hexokinase is inactive, if hexokinase is not activated, we are not going to get this reaction. We are not going to have glucose being converted to glucose 6-phosphate, which means that this right here is going to be a rate limiting step. And there's going to be 3 of those that we'll look at. Okay. The next step in the process, glucose 6-phosphate. I'm going to give you guys the enzyme, and you guys can tell me let me pull up the. I'm just going to give you guys the enzyme, and I want you to try not to look at any of your notes, and try and figure out what the next thing would be in the process without looking at any of your notes or anything, okay? So glucose 6-phosphate then gets acted on by a phosphoglucose isomerase. Anybody tell me what the next intermediate is after glucose 6-phosphate?
Fructose 6-phosphate.
Fructose 6-phosphate isos and isomerase. All we did, this exact same chemical makeup. All we did was make it rearrange it a little bit to then become fructose. Okay, so now we have fructose 6-phosphate. We have a phosphofructose kinase. Kinases, again, what do kinases do?
Add phosphate.
They add phosphate. So what is our next intermediate?
Fructose 1,6-bisphosphate.
1,6. Bisphosphate, or you can literally call it biphosphate. It's easier to remember. Bisphosphate, bisphosphate, whatever. So taking a pause for a second here, these two kinases right here, they're phosphorylating the glucose, and the fructose 6-phosphate gets phosphorylated. Where are those enzymes getting that phosphorus from?
ATP.
Exactly, from ATP. So these first couple of steps, we actually call the priming phase, because we are actually using ATP. So we're using energy to prepare the glucose to allow us to make more energy. So these two kinases, our hexokinase and our phosphofructose kinase, both use an ATP. So the priming phase, did somebody have a question? I heard somebody unmute.
I was going to ask, so every time we see a kinase, should we automatically assume that we're going to be using an ATP?
Don't assume that you're using an ATP, because the kinase is just for transferring a phosphate group.
Okay.
It can go the other way, where we're actually taking an ADP and we're taking a phosphate. We have a kinase, but that's taking a phosphate away from something and giving it to an ADP to make ATP. You can assume with that either you're using ATP or you're creating ATP, is what I would say. Or, yeah, I would say that if you see a kinase, you're either making ATP or you're using ATP, which is then why it's important to remember what of the reactions, like the order of the reactions. So if you know it's at the very beginning of glycolysis, within those first, either the hexokinase or the phosphofructose kinase, you'll see this abbreviated as PFK all the time. Dr. Sarkar loves to ask questions about PFK. Absolutely loves to ask questions about PFK. So if you see it's at the very beginning, assume that the ATP is being used up, but then at the end, we actually generate and create ATP. So.
Okay, that makes sense. Thank you.
Okay. So another quick note about this PFK, and also comparing it again about our hexokinase. Our PFK is going to be another rate limiting step. And who in their own words can tell me what a rate limiting step is?
Is it like just basically saying that like it can be inhibited or stimulated?
Exactly. It's the point of control for the reaction. Because if we think about reactions, if a reaction just happened all the time, nonstop, for example, if glycolysis happened all the time, nonstop, either we would have a ridiculous amount of pyruvate, or if there wasn't enough oxygen in the area, we would have a ridiculous amount of lactate or lactic acid buildup. But both of those things, both sides, would be incredibly bad. And then we would never have any glucose storage. So we would never have any stored energy to be able to call upon if we needed it later. Okay. So there's one more step that we consider, or technically two more steps that we consider as part of the priming phase. And that is the fructose 1,6-bisphosphate gets acted on by an aldolase. So aldolase is a little bit different. It's not one that we've talked about a lot. But an aldolase, really its job is going to be to split the fructose. So it cuts the fructose 1,6-bisphosphate. Who can tell me how many carbon molecules or carbon atoms are in one fructose or one glucose?
6.
There are 6. And does anybody know how many there are in each pyruvate?
3.
The very end of 3. So we need to go from 6 to 3. So at this point in the equation, aldolase is going to cut the fructose in half to get us in the process of eventually getting to pyruvate. It takes 1,6, so we have 1,6 carbon molecule and converts it to 2 3 carbon molecules on its way to pyruvate. Now the 2 that it creates, these are the ones that have the really, really long names, which we tend to abbreviate. So it's DHAP, which is dihydroxyacetone phosphate. And then you can also have, it's the G3P. Let me make sure that that's how he abbreviates it. Yeah, our glyceraldehyde 3 phosphate. So we get a DHAP, dihydroxyacetone phosphate, and a glyceraldehyde. In reality, though, we just want 2 of the glyceraldehyde 3 phosphates. So there's sort of it's counted as the 5th step in glycolysis, where DHAP immediately gets converted to G3P so that we have 2 G3Ps. We count it as another step, and that is through a hydrogenase enzyme. He doesn't ask a lot of questions typically on this step, but it's really important to understand that we don't immediately go straight to a G3P. We have this extra sort of side step that happens with the DHAP. Any questions or confusion up to this point? Okay, so finally now, we get into our payoff phase. We did all of this work prepping and priming our glucose to get to having a glyceraldehyde 3 phosphate. And I'm only going to show you one side, but understand that this is happening twice for each glucose. I'm just going to show you the what happens with one of them? Oh, real quick, I messed up. This is not a hydrogenase. This is an isomerase enzyme. It's a DHAP isomerase. My mistake. Okay. So with our glyceraldehyde 3-phosphate, it gets acted on by a glyceraldehyde phosphate hydrogenase. Does anybody remember what hydrogenase enzymes do? What their function is?
Do they remove water?
So a hydrogenase, it does have to do with water. You have hydrogenase or, yeah, a hydrogenase reaction or a dehydrogenase or a condensation reaction. The hydrogenase reaction is taking water and putting water in to actually break bonds and to separate things. So in this step, we have the creation, or so glyceraldehyde 3-phosphates are converted to 1,3-bisphosphoglycerate, and we create an NADH. This isn't like this is as far as we'll go with NADH right now. Eventually, NADH becomes really, really important when we get to the electron transport chain, which is after the Krebs cycle. So we have glycolysis, creates pyruvate. Pyruvate feeds into the Krebs cycle. The Krebs cycle does its whole thing, and it creates NADH, and then NADH eventually goes to the electron transport chain to create a bunch of ATP. We just need to know right now for this part that an NADH is created right here. Later on, eventually, we'll get into the actual function of that NADH and what it actually does and why it's so important. But the hydrogenase literally is going to break a bond and take off the hydrogen and add it, and is going to add it into the NADH. So the NADH, it goes from NAD plus to NADH. Does anybody recall what the coenzyme is that makes NADH? This is really pulling back some knowledge from way back when.
Isn't it like magnesium? Is it magnesium?
So if you remember coenzymes and cofactors, right? A NADH is a coenzyme. Cofactors are our metals, that sort of stuff, which all of our metals are cofactors, but our coenzymes are our vitamins. And NADH, the N stands for niacin. Can anybody remember? And niacin is just a fancy word for a specific B vitamin. Does anybody remember what B vitamin it is? It's going to be our B3 vitamin. This is probably something that in the back of your minds has kind of escaped. But the B vitamins are going to be very, very, very important later on down the road. So remember, there was that mnemonic that I gave you guys. That was, oh, shoot, how did it go? I'm forgetting it right now because it's been a little bit since I reviewed it.
It was like tall, rich.
There's tall, rich, nudists. played pickleball for centuries. And that's B vitamins 1, 2, 3, 5, 6, 7, 9, and 12. It might seem kind of like that's in the past for you guys, but I promise you this comes back really, really heavily in some of your physiology coursework, systems pathology, which is in Tri 4, clinical nutrition, which is also Tri 4, as well as boards. You will have questions on on your boards about B vitamins. If you don't remember your B vitamins, I would highly suggest to either tonight or tomorrow, sometime this week, to go back and review them just briefly, because like I said, they will keep coming back over and over and over again. And if you take a little bit of time right now to review them, you'll save yourself a lot of headache over the course of the next 2 years. Okay. Back to biochemistry. So our glyceraldehyde 3-phosphate, we have our hydrogenase, and we get 1,3-biphosphoglycerate. Now we have our 1,3-biphosphoglycerate. Going to get acted on by a phosphoglycerate kinase. Look at there, another kinase. So we talked about kinase, right? It has to deal with phosphorylation. So what do we think is going to happen with our 1,3-biphosphoglycerate? And I should clarify, kinase doesn't necessarily always phosphorylate the thing that the reactant that we think it does. Its function is to transfer a phosphate group. So it could be adding a phosphate group, or it could be taking a phosphate group away from something. So we end up with a 3-phosphoglycerate. And we know it's a kinase, so we're dealing with a phosphorus or a phosphate. And where does a phosphate within this cycle, where do we get the phosphate from, and where do we end up putting it back?
It's making an ATP, so you're adding it to ADP.
Sweet. There's our first ATP made. So then we end up with a 3-phosphoglycerate. Our 3-phosphoglycerate gets acted on by a phosphoglycerate mutase. We haven't talked about a mutase yet. So what does a mutase enzyme do?
It just moves it to the second carbon.
Yep. Yeah, the mutase, it's not going to rearrange the whole thing like an isomerase would, but it's just going to kind of pull something off and move it over to another spot. So it moves it to a 2-phosphoglycerate. Now our 2-phosphoglycerate, there's an enzyme called an enolase. Enolase is another one that you're going to really want to remember that he'll throw in there a couple of times. It's not a rate limiting step, but it is one that he likes to ask about. He likes to talk about enolase just because it's one that it's like the one step before the end. And so he likes to ask questions about that one. And this, we finally end up to phosphoenol pyruvate. So close to our goal. We have a pyruvate kinase, which deals with the phosphorylation. It takes off the phosphate group, and we end up with pyruvate. And because we took off a phosphate group, what else do we get?
An ADP or ATP.
Sweet. So the things that we get an NADH. We get an ATP here. And we get another ATP here. So there's something called like the ATP math of glycolysis, where we use 2 ATP and then we create 2 ATP per glyceraldehyde 3-phosphate. And that gives us a net total of 4 ATP plus an NADH, which later on will give us, matches out roughly to 2.5 ATP down the road. So this is something that he'll word questions like this, specifically trying to trip you up, because board says it this way. Look for the way that the question is going to be asked is, how many ATP are produced during glycolysis versus how many net ATP are produced during glycolysis. Does everybody see the difference between these two questions? My bad. Does everybody see the difference between these two questions? Where the total number of ATP that are produced during glycolysis is 4 ATP are produced, but the net ATP is 2 net ATP are produced. He will have questions like this on his exam because boards, I don't know if you guys know this, Dr. Sarkar actually writes questions for board exams. And so these sort of questions will pop up here and they will pop up on boards. I actually had this exact question on my board exam that I took to beginning of last try. It was literally this almost word for word question. He'll also give you questions asking about like in general, the whole thing, talking about how many ATP are produced in glycolysis from one glucose molecule. I also say how many pyruvate are produced in glycolysis from one glucose molecule. Or you could ask, switch it up. Let me just change this number from 2 or from 3 or from 4 glucose molecules. And so on. And just understanding that there are what is actually being produced in each one. Could also give you the question, how many NADH are produced from one glucose molecule? And there's 2. Or how many NADH are produced from a single glyceraldehyde 3-phosphates converting to 1,3-bisphosphoglycerate. And then that answer is 1 NADH. So you see, you can word these. They're all of these questions are asking pretty much the exact same thing over and over again. It's just slightly worded slightly different. It's all talking about the exact same process, and if you understand that process, you should be able to answer each of these individual questions. It's just asking them all in different ways, which is fun. The fun of test taking, of figuring all that out. So read very carefully, because he will also throw in questions like, instead of, as an answer choice, instead of 1,3-bisphosphoglycerate, he might have like 1,6 or 1,5 or 1,4-bisphosphoglycerate as an answer choice. Can anybody tell me why 1,4-bisphosphoglycerate is a completely absurd answer that doesn't make any sense?
Because there's only 3 carbons.
Exactly, because there's only 3 carbons. If there was a 1,4, then you'd have a 4 carbon structure, and that would not be bisphosphoglyceride. Cool. Last thing, in the conversion of the pyruvate, this is another enzyme that you want to remember, the pyruvate kinase. This is another one of our rate limiting enzymes. He will, I promise you, he'll have the question, which of the following are rate limiting enzymes in glycolysis? Or you just have to read which of the following are rate limiting enzymes. And he'll have answer choices A, B, C, D, and probably like A and B are correct, B and C are correct. All of the above are rate limiting enzymes. Something like that. So please, please, please remember which ones are our rate limiting enzymes.
Are those the irreversible ones?
Yes.
Okay.
Typically those are the ones that are irreversible. I don't remember off the top of my head exactly which ones are reversible and irreversible. It does say for each reaction, each step in the book, which ones are reversible and irreversible. I would highly recommend making a chart of all of these that goes over, like make a chart that includes the enzyme, reversible or irreversible, and also if it's exergonic or endergonic, because he'll probably throw questions in. He remember him he did throw questions in talking about each of those as well. And there's some general rules. You don't have to like memorize each one individually. Like if there are only three that are reversible, remember which ones are reversible and know that all of the other ones are irreversible. Or if there's if there's only 3 that are irreversible, know which those 3. If there's only 1 or 2 that are exergonic, then assume that the rest are endergonic with what it says in the book. And if you remember exergonic, look for bonds breaking, and endergonic, look for bonds forming. So making ATP, using ATP. Those are some general rules that you can follow. And in the book he has all of those things. Yeah. I did find a link. So varsitytutors.com, they actually have a lot of they're kind of like really, really short. There's only like a couple questions in each of these individual things, but they have a lot of practice tests and practice questions. Most of them are only like 1 or 2 questions each, but there's a whole bunch of different biochem topics that you can go in there and look at some example questions and kind of test your knowledge. It's already 5 o'clock, so I don't think we're gonna have time to go through all of these right now. But like I said, I will go ahead and post this as a file for you guys. I'll actually do that right now. I'll post this as a file so that way you guys can take a look at it, and I recommend going over these practice questions.
Where are you posting it at?
I'm gonna upload it onto Teams on the whatchamacallit, on the online tutoring page. Let me.
I don't think I'm part of that page. How do I become?
Are you on the GroupMe?
Yes, I am.
I'll go ahead and I'll just put it on the GroupMe right now. Is that easier for everybody?
I'm in the GroupMe of like my class, right? That's how I get onto these tutoring things because you posted.
Yeah.
Yeah.
I'll go ahead and post it in the GroupMe, and that way everybody can access. So I'd recommend go through these questions, answer them. Not all of the answers. I have the last 10. I have the answers down below, but these other ones. Sorry, one sec. What, hun? I know. Yeah, I'll go ahead and I'll post this and so that everybody can see it. And then just go over these a couple. If you have questions about the the answers there. If you can't figure out the answer, shoot me a message and I'll let you know. Okay, I gotta head out. Thanks everybody for showing up. Really appreciate you guys. And if you need anything else, please just reach out and let me know.